Good evening, everyone. My name is Dr. Stefan Ramrup, and today we'd be continuing our talk on central nervous system embryology, this time by looking at the embryology of the spinal cord and the vertebral column. This presentation is meant to build on last week's own. So, what are the learning objectives of this presentation? Well, one, we're just going to recap quickly the early steps that are directly relevant to spinal cord formation. So that'll be the closure of the neural tube, inductive role of the notochord, and how neural crest cells migrate. We'll be looking at how the caudal neural tube forms through both primary and secondary neurulation. We're going to explore how the neural tube differentiates into functional zones with the alar and basal plates. We look at the internal organization of the developing cord, the ascent of the spinal cord, and then also look at some clinically relevant pathology. So before we look at the spinal cord itself, we're just going to revisit what we talked about last week. So going back to fertilization, the trilaminar disc forms, and then from that trilaminar disc, the ectoderm of it is going to give rise to the nervous system. Running beneath that, the notochord develops from the mesoderm, establishing the body's midline axis, and that also sends inductive signals that transforms the overlying ectoderm into neural tissue. So this marks the start of neural induction, where the neural plate will fold upon itself and fuse to form the neural tube, with the closure beginning roughly around day 22 centrally and progressing both directions, anteriorly and posteriorly, or cephalic and caudally. At the edges of the closing tube, a group of cells, the neural crest cells, will detach and migrate widely, giving rise to your dorsal root ganglia, Schwann cells, autonomic ganglia, and even meninges. By the time the brain vesicles begin to appear at the cranial end, the caudal end of the neural tube is already differentiating toward the spinal cord. The cephalic or cranial portion of the tube expands to form three primary vesicles, as we said before, the prosencephalon, mesencephalon, and rhombencephalon, while your caudal portion is going to remain narrow and cylindrical, ultimately becoming the spinal cord. Neural tube closure completes when both neuropores fuse, so your cephalic one around day 25 and your caudal one between days 27 to 28. And if you have failure of this, that could lead to major congenital anomalies such as anencephaly if anterior or something such as spina bifida if caudal, with these events being fully dependent. So within this closed neural tube, molecular gradients now begin to create the internal organization. You have signaling proteins such as sonic hedgehog secreted by the notochord, and floor plate directs the ventral or motor development, while BMP and WNT signaling from the surface ectoderm guides the dorsal sensory differentiation. The balance between these gradients establishes the alar plate dorsally for sensory structures and the basal plate ventrally for motor nuclei. So with that in mind, let's look at how the spinal cord itself forms through two processes, primary and secondary neurulation. So during primary neurulation, the neural plate folds and fuses to form a hollow tube that continues down to about the S2 or the second sacral level. And this occurs around days 22 to 26 and forms most of the spinal cord from the cervical region through to the upper sacral segments. And below that point, secondary neurulation takes over. Here, your mesenchymal cells in the tail bud condense into a solid medullary cord, and this cord is then going to hollow out, canalize, and connect with the neural tube above. This region forms the conus medullaris and filum terminale, which complete the spinal cord, and together these processes create one continuous lumen, which is the future central canal. So within the newly closed neural tube, differentiation occurs along the dorsal-ventral axis. A shallow longitudinal groove called your sulcus limitans appears in the lateral wall of the neural canal, and this divides the neuroepithelium into a dorsal alar plate and a ventral basal plate. The alar plate, positioned dorsally, gives you your sensory neurons. These neurons will form the dorsal horn of the spinal cord, and they will receive input from the dorsal root ganglia that originate from neural crest cells. And then eventually, basal plate forms motor neurons, and their axons extend outward as ventral motor roots, which eventually join with the sensory fibers to form your mixed spinal nerves. The midline structures, the roof plate dorsally and the floor plate ventrally, will act as signaling hubs, with the floor plate releasing sonic hedgehog signaling protein, while the roof plate influences commissural axons as they cross the midline. And by around week 6, this organization is fully established of dorsal sensory and ventral motor. As differentiation progresses, the wall of the neural tube organizes itself into three concentric layers, with the innermost being the ventricular ependymal zone, which contains rapidly dividing neuroepithelial cells. These cells give rise to neuroblasts, which then migrate outward into the mantle zone. The mantle zone is where the spinal cord's gray matter forms. Within it, the alar and basal plate derivatives expand, with the dorsal horns emerging from the alar and ventral from the basal. That will give you your classic butterfly-shaped gray matter pattern that we see in a mature spinal cord. The outermost marginal zone contains the axons of these developing neurons and supporting glia. As these fibers become myelinated, the zone is going to transform into your white matter. Myelination begins late in fetal life and it continues into infancy. That's also why neonatal reflexes and motor control mature gradually after birth. And the original lumen of the neural tube remains as the central canal, and that maintains continuity with the brain's ventricular system. Before the spinal nerves emerge, the neural tube must be enclosed, and that's not just by bone, but it's also by protective membranes, which is the spinal meninges. This begins around week 4, when your mesenchymal cells and your migrating neural crest cells condense around the neural tube to form your primary meninx, primitive connective tissue sheath. By weeks 5 to 8, this meninx differentiates into 2 distinct layers. The outer one, of dense mesodermal layer, becomes the dura mater, and the inner, more cellular layer of neural crest cells splits into the arachnoid and your pia mater, which are collectively called your leptomeninges. Small vacuoles will appear within the inner layer, and as they coalesce, they'll create your subarachnoid space, which will later be filled with cerebrospinal fluid. And by around week 8, cerebrospinal fluid production begins, and that cushions the cord. As development continues, your dura will extend farther caudally more than the cord itself, and that's going to form the lumbar cistern, while the pia mater gives rise to denticulate ligaments that anchor the cord laterally and the filum terminale at its base. With the spinal cord's internal architecture established, you have the next step would be the outgrowth of its connections, the spinal nerve roots. So your motor fibers will develop first, and these axons arise from motor neurons in the basal plates of the ventral horn, extending outward through the ventral aspect of the cord to form ventral motor roots. That begins around weeks 5 to 6, and simultaneously, neural crest cells migrate laterally and aggregate besides the cord to form your dorsal root ganglia. Each ganglion's neurons extend central processes into the cord's alar plates to establish sensory input pathways and peripheral processes toward the body wall and limbs. When the ventral motor fibers and dorsal sensory fibers unite, they form your mixed spinal nerves containing both afferent and efferent fibers. Each of these nerves will align with a single somite, and that's going to give rise to the segmental pattern of the nervous system and the corresponding dermatomes and myotomes that we know. So with respect to ascent to the spinal cord, in the early development, the spinal cord and vertebral column are perfectly matched in length at that point in time. Each spinal nerve exits directly opposite its segmental level in a one-to-one correspondence between the neural and skeletal axes. As the embryo grows, the vertebral column is going to elongate at a faster rate than the spinal cord. And because the cord can't keep pace, it appears to ascend within the vertebral canal, while the nerve roots are drawn downward to maintain the exits through their appropriate foramina. That stretching is going to create your chorda equina, or your horsetail of descending roots. By about week 8, the cord still reaches the coccyx. At birth, it ends around L3, and in the adult, it stabilizes around L1 to 2, forming your conus medullaris. And below that, you have the pial extension, giving you your filum terminale, which anchors the cord to the dural sac and the coccyx. So the vertebral column originates from the paraxial mesoderm, which lies on either side of the developing neural tube. As the development continues to progress, the mesoderm segments into a head to tail direction into somites starting around day 20. So each somite has 3 functional zones: your sclerotome, myotome, and the dermatome. The sclerotome will give rise to your vertebra and your ribs. The myotome, your skeletal muscles, and your dermatome forms the dermis of the overlying back. The sclerotome cells are of importance here, so they migrate immediately toward the notochord and the neural tube, and they wrap around them. Those that encircle the notochord will form your vertebral body, while those that surround the neural tube form your vertebral arch. So now moving on to resegmentation, rather than each somite forming a single vertebra, the caudal half of one sclerotome is going to fuse with the cranial half of another one. And that shift ensures that the spinal nerves emerging from the neural tube pass between vertebra through the newly formed intervertebral foramina. Meanwhile, the intervertebral discs form right where the original notochord persists, which you can see in the picture on the right-hand side. The central portion of the notochord remains as the nucleus pulposus, and the surrounding mesenchymal tissue differentiates into the annulus fibrosus, and that's going to create the intersegmental joints of the spine in a disc vertebra disc pattern. If you have any issues with resegmentation failing, you can get congenital anomalies, such as block vertebra, hemivertebra, or Klippel fail syndrome. We have segmentation defects that could lead to abnormal spinal fusion and limited motion, which we'll discuss later. So just taking a closer look at that picture I was just talking about. In panel A, you can see the sclerotum cells condensing around the notochord, which is the first step in vertebral body formation. And the notochord runs along the midline, and the surrounding skeletal mesenchyme migrates immediately to encase it. In the second part of panel B, that mesenchyme begins to organize into a metameric pattern. So that is alternating dense and less dense zones, with the more condensed regions becoming the vertebral bodies, while the less condensed regions in between form your early intervertebral disc spaces. Also notice how the notochord expands in these less dense zones, persisting as the nucleus pulposus, while it degenerates within the future vertebral bodies. The surrounding sclerotomal cells within the less dense zones differentiate into the annulus fibrosus as well. So this slide is more a summary of how the cord and the vertebral column are kind of how they coordinate in development. So it's just more or less a summary of that. So because of resegmentation, each vertebra forms slightly offset from the original somite bodies, and that allows the spinal nerves to exit between vertebra through the intervertebral foramina, which gives you your segmental organization of dermatomes, myotomes, and sclerotomes. And as the embryo grows, the vertebral column will lengthen more rapidly than the spinal cord, giving you the appearance of ascent of the cord. So now that we've talked about the normal development of the spinal and vertebral column, what happens when that doesn't go according to plan? So even with minor disturbances in neurulation, that can result in major malformations, that with some possibly being visible at birth and others that can remain hidden until adulthood. So broadly, they fall into two categories. You have neural tube defects, which is when closure fails or and vertebral segmentation, and you have neural tube defects and vertebral segmentation defects. They can coexist. So you can have a myelomeningocele with a Chiari-2 malformation or a scoliosis from vertebral asymmetry. So firstly, just looking at spinal dysraphism. So we'll be looking at spina bifida first. So spina bifida occulta is the mildest and most common form of spinal dysraphism. It doesn't arise from failed neurotube closure, but from incomplete ossification of the dorsal vertebral arches, mesodermal defect of the sclerotum. And this typically occurs at the lumbosacral junction, most often L5 or S1. The spinal cord and the meninges here remain perfectly formed, hidden within the canal, hence the term occulta or hidden. Externally, you might notice subtle cutaneous markers, so that could be a tuft of hair, a small midline dimple, a dermal sinus tract, or a subcutaneous lipoma. And most patients are asymptomatic, but these surface clues are important because occasionally there could be a tethered cord or intradural-like puma that could be underneath. So in neurosurgical practice, any midline lumbosacral skin lesion, no matter how small, should be imaged. And occult defect can still tether the cord and produce progressive neurological symptoms that could occur in later childhood. So moving on to meningoceles, they represent the next step in severity. So here the neurotube has closed normally, but the vertebral arches fail to form, creating a posterior defect. Through that gap, the meninges herniate outward as a CSF or cerebrospinal fluid-filled sac, and that's lined by dura and arachnoid. What's crucial here is that the spinal cord remains within the canal and neurological function is preserved. Clinically, this can present as a soft cystic midline swelling over the spine that can typically be translucent and covered by skin. These patients have no motor or sensory loss, but the sac is vulnerable to trauma or infection, and surgical management typically involves excision of the meningeal sac and closure of the dural and fascial defect, primarily for protection and cosmesis. And although neurologically intact, these patients do need long-term follow-up as they occasionally develop tethering or arachnoid adhesions over time. The myelomeningocele is your open neural tube defect at the caudal end, It results from failure of caudal nerve tube closure during primary neurulation, and that leaves a gap through which both the meninges and the spinal cord herniate. It almost, or mostly it occurs in the lumbosacral lesion, but you do have cervical and thoracic variants that exist. The exposed neural cord will lie at the surface, and it's often covered by a thin meningeal membrane or none at all. And neurologically, these patients have flaccid paralysis and sensory loss below the lesion, along with neurogenic bladder and bowel dysfunction. Most can also be affected by other findings at the same time. So, as I mentioned before, a KRA2 malformation, where you have the cerebellar tonsils and brainstem herniating downward, and hydrocephalus in about approximately up to 80% of cases. From a neurosurgical standpoint, early surgical closure within 24 to 48 hours of birth is important to prevent meningitis and further neural injury. And many do require ventricular peritoneal shunting for hydrocephalus and later management of tethered cord or orthopedic complications. Myelolesthesis represents the most severe failure of neurulation, and it's a complete absence of neural fold fusion. In this, the neural plate will remain open to the surface with no meningeal or cutaneous covering. The spinal cord basically, or essentially, is a flat neural cloud directly exposed to amniotic fluid, which causes progressive deterioration of neural tissue in utero. Embryologically, this results from an arrest of primary neurulation between days 22 and 26. That's long before the embryo even reaches a centimeter in length. And clinically, this can present as a completely open defect with no possibility of functional neural tissue below the lesion. So the final group of spinal pathologies involve secondary neurulation, where this process forming the distal cord and the phylum. So one, in tethered cord syndrome, the distal spinal cord becomes pathologically fixed, often by a thickened phylum terminale or a dermal sinus or a lipoma. As the child grows, the vertebral column elongates, but the cord cannot ascend normally, and that's going to lead to stretching, which can lead to stretch-induced ischemia. Clinically, that will progress as neurological decline that develops over time with progressive low limb weakness, gait changes, sensory loss, and bladder or bowel dysfunction. Surgical untethering releases the cord and halts further deterioration. Secondly, you have caudal regression syndrome, which arises when the caudal cell mass fails to form properly during secondary neurulation. The lower sacral and coccygeal vertebrae are absent or rudimentary, and the distal spinal cord may be truncated or missing entirely. This is often linked to conditions such as maternal diabetes, and it can have varying severity from isolated sacral agenesis to a complete lumbosacral absence. So moving on to vertebral column defects. So a block vertebra forms when two adjacent sclerotomes fail to segment properly, and that can result in congenital fusion of their vertebral bodies. This is as a result of failure of the resegmentation process. So the intervertebral disc never develops between them. It most often occurs in the cervical spine, where it may restrict motion or predispose to adjacent segment degeneration. Most cases are asymptomatic, but if the fused segment alters alignments, you can get nerve compression, torticollis, or even vertebral artery stretching if you have a high cervical lesion. Hemivertebrae occurs when only one half of a vertebral body forms. Essentially, you have ossification failure on one side of the sclerotome. That could create a wedge-shaped vertebra that tilts the spine, and that could produce a congenital scoliosis. Depending on whether it's fully or partially segmented, the deformity can be static or progressive. Fully segmented hemivertebra with normal discs above and below will typically worsen as the child grows, while a semi-segmented or incarcerated one, fused to adjacent levels, tends to be more stable. Finally, Klippel-Feil syndrome. It's a classic example of multi-segmentation failure involving fusion of two or more cervical vertebrae. It arises between weeks 3 and 8 when your cervical somites should separate, but they remain fused instead. Clinically, you can have a triad of short neck, a low posterior hairline, and restricted neck mobility, but only about half of these patients have all three features. Because of the abnormal vertebral motion above and below the fused levels, these patients often develop instability and degenerative changes at adjacent segments. And they also have a high rate of associated anomalies, so that includes Sprengel deformity, which is an elevated scapula, renal anomalies, congenital heart defects, and syringomyelia. from a neurosurgical perspective, the key concerns would be cord compression, vertebral artery anomalies, and um, careful planning for any posterior fossa or spinal procedures due to an altered bony anatomy. So with that at the end, with the anomalies being discussed, that brings us to the end of the presentations on central nervous system embryology. Thank you.